All right, why don't we get started? Today we have the pleasure of presenting BISC Global, an extreme bioinformatics software and consulting company. And they are going to present some real world case studies on analysis and different techniques that they're doing in biotechnology today. And they're going to start at a higher level and dig in and go into some detail. So the good news is it'll be recorded. So if you want to come back and watch it, learn more from it, you'll be able to do that. So our rules of engagement, we'll address questions after the last presentation. This way, everybody gets to give their presentation without interruption. So if you have a question, put it in the Q&A. It'll get addressed at the end. Some people use the chat box. It doesn't matter, chat or Q&A. Either one will address your questions. If you have to leave, again, you could come back and watch the recording, or just send us a note and saying, hey, wanted to follow up. You, as a general attendee, you can ask those questions, and they'll get moderated. Panelists can also ask and answer questions, and then it'll be recorded, it'll go on demand, and you can follow back up at your leisure. Today's speakers are Matthias Banz. He's the development team leader and senior software developer and cloud enthusiast at BISC Global. Emil Gerards, bioinformatician at BISC Global. And then Dr. Alexander Luganov. He's the scientific director of machine learning and AI, uncertainty analysis and quantifications at BISC Global. Matthias's talk is going to be scalable and secure big data cloud solutions and environments. Emil's is going to be uncovering master regulators using single cell transcriptomics. And Sasha's is going to be machine learning and AI and protein engineering applications. And so from here, I would like to hand it over to Matthias.
All right, thank you, John, for the introduction. I hope I am clear, loud and clear for everyone. All right, perfect. Sure. I'm going to share my screen. Perfect. So hello, everyone. Good morning, good evening, depending on which side of the pond you're located. So I'm Matthias Banz, and in the following presentation, I'm providing you an overview of scalable and secure high-performance cloud computing architectures. So pharma and biotech are changing gears. Predictionist models are replaced by system models based on massive amounts of data. By integrating different data dimensions, we can, for example, model complex phenotypes and more. We place disease mutations, genes, transcripts, and more in biological context, which help us to identify the best data points for assessing disease risk and therapeutic intervention. However, biologists are generating data on a scale that is outpacing our ability to perform even the simplest analysis. Sequencing from whole genomes to thousands of single cells to imaging data of thousands of histological tissue slices are all growing at a phenomenal rate. Analyzing those data and integrating them to form a more complete picture of biology will require access to large-scale and affordable computing. But first, let's look at the type of computational challenges currently facing big data researchers. As an example, let's imagine a genetics lab trying to identify cell populations and associated dysfunctional gene regulations involved in the progression of disease, diabetes type 2 via a single cell transcriptomics study. They will identify transcripts in the RNA sequences for several thousands of individuals with type 2 diabetes and their controls. In this scenario, each individual generates tens or even hundreds of gigabytes of raw data. The problem occurs once researchers wish to analyze and integrate their information about transcripts with known pancreatic cell compendia into cell identities and differential expressed transcripts. A uniform manifold approximation and projection, for example, shows how transcripts and cell populations may causally be related to another to produce a type 2 diabetes phenotype. And this is where they encounter another computational bottleneck. For only limited transcripts and cells, the number of possible combinations would be trivial, but the number of possible combinations skyrockets to 10 to the 18th power, and transcripts and cell amounts get bigger. The computational power needed to tackle these analyses grow exponential. The resulting long computational latencies can severely slow down research or even diagnosis in clinical settings. Over the past several years, major innovations have provided scientists with more effective and cost-efficient methods for high-performance computing systems. Such one is cloud computation. Cloud solutions make data and computational resources available to departments, a whole company, or even external collaborations. Of course, you should be aware of the difference between on-premises and cloud-based computing to make a better decision. When it comes to scalability, you pay more for an on-premises setup, and you also have lesser options. Once you scale up, it's often difficult to scale down again, and often leads to heavy losses in terms of infrastructure and maintenance costs. Cloud, on the other hand, allows you to pay only for what you use, and easily scales up and down if required. Next, data storage. On-premise systems need a lot of space for their service, power, and maintenance. On the other hand, cloud providers take care of this by their own cloud storage solutions, so you save on maintenance, time, and hardware material. Then we have data security. On-premise systems offer less data security thanks to a complicated combination of physical and traditional IT security measures, whereas cloud computing offers much better security if set up correctly and lets you avoid having constantly monitor or manage security protocols. In the event that data loss occurs, the chance of full data recovery on on-premise setups is very small. In contrast to cloud computing systems, they have more robust disaster recovery measures in place to ensure faster and easier data recovery. And finally, we have maintenance on-premise systems also require additional team for hardware and software maintenance, loading up the costs by a considerable degree. Cloud computing systems, on the other hand, are maintained by the cloud providers, reducing costs and resource allocation substantially. I have selected 2 example projects where BISC Global teams combine their biological knowledge and cloud expertise within cloud solutions. My first example is OP², our online pipeline platform, which shows our capabilities as a company. And this online user-friendly, secure, and scalable platform offers scientists the possibility to analyze omic data sets with the ease of a few clicks without any prior bioinformatics or cloud knowledge and expertise. The software as a service framework has been developed by our multidisciplinary team. Our cloud engineers built and deployed a serverless, instant, scalable, and data secure environment for compute and storage. Our biology and omics domain experts consolidated their NGS, next generation sequencing knowledge, into scalable pipelines that follow the latest changes in the field. On the other hand, our application developers created interactive insight reports of the complex data built on a clear user interface. My second showcase is the cloud infrastructure we have set up for a university hospital. The request was clear. Move their clinical whole exome sequencing pipeline to the cloud. We migrated their logic to the cloud, environment they selected. The environment auto-scaled up and down depending on the sample load and the urgency for diagnosis. We have optimized their sequence pipeline and applied code versioning. We built a highly secured Bastion cloud environment within their private subnet and applied data lifecycle policies for cost-efficient, long-term data storage. Within the current setup, the hospital only pays for what they use, and they have reached a 4 times faster sample turnaround at lower costs. Now, I would like to address 3 popular, frequently asked questions about cloud computing I get during my project scoping meetings with our clients. The first one is, how is data security handled in the cloud? Virtual machine instances behave the same as physical machines. It is up to the user to restrict access to the machine using firewall and encryption settings. Open ports should be limited to those necessary for function, and all data and communication should be encrypted. This is also a responsibility that BISC takes up during the project. The second one is, how is data transfer handled in the cloud? Data transfer speeds are limited by the speeds of the internet connection between you, the user, and the cloud service, of course. As such, transferring large amounts of data might be troublesome. And for this purpose, cloud providers offer solutions for a fast, direct connection to their compute resource. And thirdly, does data persist in the cloud? Cloud data is stored on a network storage system. The user's virtual machines pull stored data from this system and write results there as well. While data placed on or generated by virtual machine image only persists for the lifetime of that instance on the network storage system. And also for this, we have multiple cloud services we can implement in your cloud environment. This concludes my presentation, and I'm Matthias Bijns. Please do not hesitate to ask any questions now, or let's say after the webinar, of course, to me or to one of my colleagues. One of my colleagues is Emil, he will introduce you to the wonderful world of single-cell transcriptomics. Emil, the floor is yours.
Matthias, I'll share my presentation now. It should now be visible to you. Shout if it isn't.
Looks good.
Good. So I'll talk about uncovering master regulators using single-cell transcriptomics. So today we're going to dive deep into single-cell sequencing and gene regulatory networks. Don't worry, I'll introduce both topics first, after which we'll dig deep into one of my favorite tools for this kind of analysis. It's called Scenic. I will end with 2 real-life case studies where we applied Scenic to help clients. So first, as promised, single-cell sequencing. As the name implies, you have to first generate your single cells. This is usually done by dissociating your tissue into a solution, and then either pipetting a single cell into a plate or using an advanced microfluidic device to generate oil droplets containing just a single cell. Once you've isolated your single cells, you can start extracting and sequencing their RNA molecules. After that, you can map that to the transcriptome, and then you'll end up with what's called a gene count feature matrix, or count matrix for short. This is a very large table where the columns represent the individual cells and the rows represent the individual genes. And so for each cell, you have the measured amount of mRNA molecules for each gene. As you can imagine, this table is very large. Hence, your data analysis, or one of the key aspects of the data analysis that I want to highlight today, is the dimensionality reduction that takes place. So we have a high dimensional matrix of around, let's say, 26,000 genes. I plotted here 3 for convenience. Any more than 3 would make your head hurt. So how do you collapse, let's say, this matrix into those nice T-SNE and UMAP plots as shown by Matthias or shown in publications? Well, the answer is dimensionality reduction. It's going to be done in multiple ways, but most often scientists extract the top 3,000 to 5,000 most variable genes, run principal component analysis, and based on that reduced dimensional set of, let's say, 30 principal components, that's what they use to create their visualization. I can talk much more, but I'll move to gene regulatory networks in a bit, after I've highlighted that the sequencing and mapping step and the data analysis step, if they sounded very complicated, is also very much within scope for our OP² platform that Matthias highlighted, and it can take care of all these complicated steps for you in just a few clicks. Now, gene regulatory networks are often associated with these very complex hairball-like figures. They have a lot of dots and a lot of arrows. Again, I'm not going to dive too deeply into this topic. I'm just going to highlight two main points which are fundamental to any gene regulatory network, and that's how this method or each method selects their co-regulated genes. And so each dot, why does it group these dots in the way that it does? And the jargon is called how do you select the nodes. Then your method also has to account for the control of each gene set. So how do the genes regulate each other? This is represented by the arrows, or as they're called in the jargon, the edges. So each method has their own way, and the use of prior knowledge is a key aspect. The use or not use of prior knowledge is a key aspect here. Methods can use very small, very limited prior information, so for example, just some genomic information, up to fully validated protein-protein interaction databases to create this kind of network. Of course, you have to take into account that the more experimentally validated model you want to start from, the more certain you will be of the interactions or the predictions the model provides. However, it will produce very little novel models or novel interactions, and interactions that were not known before. So there is a balance, let's say, and you have to strike between what you want to find novel interactions or you want to find interactions that are most likely biologically true. In addition, something to take into account as well is that prior knowledge may not be generated for the biological system, so the species, the tissue, or the cell type that you for your experiment are looking at. So this is a very important caveat when creating, interpreting, and using these kind of gene regulatory networks. All right, this concludes the introduction. Now let's move to my favorite tool to do gene regulatory network analysis in single cell sequencing. So the tool is called scenic. It has three main steps, and they address the key points that I highlighted first. So again, how does scenic collect the co-regulated genes? How does it connect, let's say, the transcription factors to the genes? To do this, it uses a random forest regression model, so a machine learning model, to infer co-regulation between transcription factors and expressed genes. So it takes in your count matrix, so your cells and your genes, and it looks for gene modules, so genes that are co-expressed and are significantly associated with a specific transcription factor. So this first step creates or investigates correlation, co-expression. Second, scenic adds in the causal aspect of your network. To do this, it uses a rather minimal amount of prior knowledge. It just uses the genomic information of your expressed genes. So it checks whether the genes in your predicted modules share a motif, so a genetic sequence that corresponds to the binding site of the predicted transcription factor. This is really a sanity check. can the transcription factor that I think regulates these genes, does it have the actual ability to do so? Is there the potential? Doesn't mean that it's actually doing that, but can it do that? So based on that, your regulons or your groups of genes get pruned. So only genes that effectively have this kind of the correct motif are retained, while others are discarded. So we end up with what CINIC calls a regulon. So that's a group of co-regulated genes that share a transcription factor motif. So that's the causal aspect. Then how does it use these regulons? It again creates a regulon activity matrix. So for each regulon, it will check in each cell how active is this regulon. And this activity corresponds to the joint expression of all the genes in those regulons. If you do this for all genes and all regulons, you get, again, a matrix, which is quite similar to the count matrix, the rows being the cells and the regulons, and the regulon activity scores being the rows. So these are the main outputs of your CINIC tool. So this is the regulon activity matrix. For each cell, you get the relative biological activity of your regulon. And you can use that to create summaries per cell group, for example, per cell type or per condition, to see which regulons are most active and which are the master regulators, which are the determinants, let's say, of your cell phenotype. I'll show examples in the case study of this. Now, you may have noticed that this type of matrix is very much reminiscent of your count matrix, only it has much fewer dimensions, only a few hundreds compared to your full transcriptome. So you could also use your regulon matrix or your regulon activity matrix for visualization, downstream analysis, anything you can do on a count matrix. You can also, or you can use your regulon matrix also as dimensionality reduction. In addition, apart from this regulon activity matrix, it also provides you with a de novo predicted regulatory network. De novo, because remember, your co-regulated genes are really created based on the data you put in and nothing else. So it's just based on your expression matrix. So you know it's specific for your animal, for your species, for your cell type. So you know that you are working with a very new, very novel network. And it's very interesting to look, for example, for the effector genes and which genes are mediating the phenotype, and are there any interesting targets or modulators within that set. So to summarize CINIC produces regulons and de novo predicted sets of co-expressed genes with a shared transcription factor. Now on to the case studies. So the first case that I would like to highlight is a differentiation process where we were looking at the oligodendrocyte differentiation in the brain. This is very relevant because diseases of the brain that includes remyelination are severely hampered by our lack of understanding of how these oligodendrocytes reproduce and differentiate. So here, what we did is we actually leveraged public, already available single-cell RNA repositories. We combined a trajectory analysis with this network inference. So we plotted, or we created in silico life cycle, let's say, of your progenitor oligodendrocyte precursor cell to your fully mature myelinating oligodendrocyte, with then the objective to find what is driving this differentiation. How does it look? So what you have here is a summarized regulon activity matrix. It shows the average activity of a set of regulons, being the rows here, for each cell type or each cell group, here being the columns. And as you can see, we could focus this analysis nicely on intermediate cell types. So when they are starting to differentiate, what is driving this differentiation? What are the key transcription factors that mediate this, and what are the key genes that are responsible, the key proteins, let's say, that are being transcribed here. So we could really focus on the master regulators of differentiating cell types using this scenic analysis. The second case is where we had a disease process and the client was very interested in the effects of the compound or a treatment on the disease process. In this case, we were talking about fibroblasts. So in fibrosis, there is really a excessive formation of what's called myofibroblasts or inflammatory fibroblasts. Here, the client had a very large data set available with multiple compounds, and we performed network inference using scenic on the total fibroblast population. And so it's a bit more complicated here, but you can already appreciate that you can really use this technique to identify regulons that are both specific to each given cell population, or also, for example, things that scale up during the fibrotic process. And you can really use this type of information to find the switches, the master regulons that drive this fibrotic process. In addition, you can do the same just by grouping or averaging across your compounds. And you see there is a very dramatic difference between regulon usage in your disease and in your control condition. And you can also already appreciate that, for example, that adding a compound attenuates this regulon signature, not fully, but okay, this is very clear that there is a quite promising effect of the compound. So your scenic can also identify disease master regulators as well as focusing on the compound effects. That was my last slide. So I hope I left enough time for you, Sasha, to take it away, because you have a wonderful slide deck on machine learning applications.
Thank you, Emil. Let me share my screen. Can you see my screen?
Yes.
Okay, thank you. Well, good morning, good afternoon, depending where you are. Thank you for your time. And today I would like to talk a little bit about the machine learning AI. I guess everybody knows that it at the moment has a lot of application across different industries and domains. And I would like to talk today about application of machine learning in AI and protein engineering. The way I will structure my presentation, I will firstly dive into the one specific example of our work recently we've done, which is related to protein engineering. And then at the end of the presentation, I will show you a couple of additional applications where machine learning AI can be used successfully and you can get very interesting results. So the subject of my talk today is CFI protein engineering with application of machine learning. So first of all, let me talk a little bit about high-level review, what was the project is about. So the goal of the project was obviously to look at the impact of the changes in amino acid sequences and impact of those changes towards the behavior and properties of the protein. Obviously, ideally, we would like to find interesting mutation, which is targeting particular disease or particular properties, and to build a large library of variants, which can be used and tested in the lab facilities and speed up that process, because in all our circumstances, it will take a lot of time to generate those libraries. For that reason, machine learning and artificial intelligence plays a significant role because it allows actually to construct the necessary pipeline and prediction algorithm, which leads eventually to the set of sequences you can use in your study. Obviously, there are multiple aspects which involves into the building machine learning. And in this case, we rely on some of the initial data. It could be just sequence data, or it could be also 3D structure of the protein, which can be used to incorporate in the resulting machine learning approach. So the benefits of using the deep learning, because you obviously can rely on your domain knowledge and your biological intuition, but most of the time machine learning allows you to identify the area and some de novo type of residue where you can introduce the mutation to meet your desirable goal. And that's where the beauty comes with the application of machine learning. So let me talk a little bit about the CFI protein, or complemented factor 1. So it is known as a C3b or C4b inactivator, and it is a protein within the human encoded by the CFI gene. Usually it is soluble and it's circulated in a human bloodstream. Most of the time it is expressed by liver, but it's also expressed by in some other locations, and it contains the heavy chain part of the chain and also the light part of the chain. And obviously that plays a very important role in multiple signaling pathways you can see on the slide. And our goal is actually trying to characterize the protein and come up with a potential modification of that protein and engineer the sequences which will lead to desirable properties. So as I mentioned, this complement factor 1, basically a protein which complements the system and regulate the complement activation by cleaving cell bound C3b and C4b. And for that reason, obviously, the assay can be built to monitor C3b and C4b cleavage. That's usually done by building a necessary system, and via incubation process, you can detect formation using, ELISA technique type of things. So what kind of data we can get as a result of that? Obviously, we for the different assay, you can measure certain parameters of the assay, but also you can include the sequence data for the particular situation you are interested in. And also you can measure C4C screening activity and also C4C AC50 activity, and that's the same applies to the IC3B screening values and IC3B AC50 activities. There are two venues here to follow up in terms of the application of machine learning. So we can, based on frequencies of appearing certain amino acid in the sequence, and that would be our input in the machine learning. But also we could apply on the whole structure of the sequence and the position of amino acid in the sequence, and then build the model which takes into account the structural aspects. So we actually decided to follow up with the structural aspect, and the reason for that, because we want to identify the position of the de novo residue, which leads to where we can introduce the mutation and which leads to the desirable properties. And in order to achieve these things, we can apply coding of the sequence in terms of the matrix. That's usually done by one-hot encoding process, where you have the sequence and then the sequence is coded into the table or matrix, let's put it this way, as you can see on the slides. And as a result, you will get about 2,400 let's say tables, depending on your training set. In our case, we have about 2,400 sequences. And this forms is our training set for which we know activities, and we can build the machine learning model further down. The way we decided to tackle that problem, not necessarily related to the regression aspect, but we decided to do the classification, meaning that the outcome of our prediction will be AC50 activity, which will be in a certain range. So that problem becomes more classification problem into the appropriate ranges of AC50 activities, rather just doing direct regression on the values. And it's more stable and robust. The way we design our usually machine learning architecture obviously depends on the problem you have in hands. And obviously we apply a lot of optimization on the top to show that we actually build very optimal structure for machine learning in order to achieve certain results. For that particular application, it had two components, obviously, one which comes before the compression layer, which requires also one-hot encoding type of step, but also the compression layer, which actually squeeze all those huge set of matrices into something more coarse, and I would call those coarse features. And those coarse features are used to train the model using the number of hidden layers and then predict and produce their output. So this is the general structure of the neural network. So as a result of that activity, we build our own neural network. We also compare with the published in the literature some of the machine learning approaches, and also we compared our machine learning with respect to just naive convolutional neural network. And we see that the approach we choose as our default approach has a better sensitivity and selectivity for classification problem, and gives us a confidence that we can make appropriate detection of the residue for the mutation, and also make a future prediction and filtering the sequences which meets the target. So very actually interesting for the high EC50 activities and a mutation which leads to high EC50 activities. Okay, so this is the general structure of the CFI protein. As I mentioned before, it has a A chain heavy one and a B chain light one. We were particularly interested to make the mutations in serine protease domain, SPD domain, but we also look at the other domain, mainly in the A chain and the de novo residue, which potentially may lead to high EC50 activity, and that's something out of the scope of the conventional consideration by the biologists or protein engineers, but it still provides some insightful information in terms of where you can also put some of the mutations. And there are two approaches here. One is, as I mentioned, is a knowledge-driven biological selection, meaning that you know that there are some loops in SPD domain, which does play the important role in activity and the binding properties. So, and then in those are loops, for example, you know, particular residue, and then you can try to mutate about those residues and produce possible variants of your sequence of the original protein. And that's one of the approach. And the second approach is purely based on the machine learning driven biological selection. Then machine learning tells you those are residues sensitive towards the producing modification in AC50 and suggest you to mutate around those residues. And then obviously you need to run the second time machine learning to filter out the sequences which leads to decreasing the EC50 activity versus to increasing, and only keep one which leads to increase of the EC50 activities. So, but the question is still there in terms of how far you can go from those residues and what you need to mutate. And from that point of view, obviously we need to bring 3D structure of the protein into the game. We need to look at the distance matrix between amino acid and the 3D structure. And then we need to look to neighborhood of the particular residue of our interest. And we decided to focus on the distance about 7 angstrom. This allows us to limit what we can mutate and produce the limited number of the sequences for further considerations. So once we have formulated all those aspects in place, the remaining questions is actually what we're going to substitute at the position we identify as a possible mutations. And there are obviously a lot of combinations if we're talking about all possible scenarios across the 20 amino acids. And for that reason, we decided to focus on only 3 mutations around the possible residue. And also we look at the substitution matrix and we choose the unlikely mutations between amino acids instead of more likely mutations. So we are looking to that matrix and we picked up unlikely mutations and then make substitution in the residues and position we identify either from the knowledge-based approach or machine learning-based approach. So maybe a few words I need to say in terms of compression. That's a very important step. As I mentioned, you can, of course, build the machine learning using the original one-hot encoding sequences, but it's a lot of data and requires a lot of memory, and computational time of training and getting actually pretty good results takes a bit of time. So therefore, compression into the more important features which still contains the necessary information and train on those features is a game-changing technology, a step here, because it allows you much faster generate your neural network, optimize that neural network, and make some sort of discovery of your residue for mutation, and also afterwards the filtering them out in terms of showing that they actually leads to high EC50 activity. And for that reason, we use the compression layer. Most of the time it's a modification of autoencoder, which kind of allows you to compress and decompress information. So we're compressing the set of the matrices into the much smaller subset, and they use this one for the training purposes. And just one slide. So when you compress the data, obviously you have, you lose some of the information. And when you decode it, you could have some artifacts as a result of that process of encoding and decoding. And it's very important to maintain the high quality on both ends when you compress and decompress information. And in terms of the feature importance, when we have a compressed information in our system and we train our machine learning model, so the next step obviously would be important is how we build the algorithm which actually identify the position inside the sequence which needs to be mutated. And the way we constructed that process is based on the first of all identification, what are the most important features which is sensitive towards the changes of AC50. And then we decoded that feature into the original matrix. And then as you can see, position highlighted by green on the slide, which leads to the position in the matrix. And then those are position converted into the position in the sequence, original sequence we used to use to generate the matrix. And that's what leads us to the information where we need to introduce the mutation. Then we can use the distance matrix to make the mutation subject to the substitution matrix. Okay, so as a result, we have an algorithm and pipeline, and we were able to build based on the knowledge or domain expertise, the set of the mutation, but also we were able to identify very highly likely candidate residuals where we need to introduce the mutations. So we select 3 out of 14 important residues, and then we generated quite a big substantial set of potential variants of the sequences, which leads to high AC50 activity. And at the end of my presentation, I want also to highlight a couple of other applications. So we also done very successful project where we applied the machine learning for prostate cancer diagnostics. So we use the set of the biomarkers and develop sophisticated machine learning model to make the classification very accurately. And that's, became as one of the possible substitution of the conventional PCI type of test, and it leads, much better sensitivity, specificity, and diagnostic between less aggressive cancers and more aggressive cancer. Another application obviously related to studying and prediction of thermodynamics properties, so monoclonal antibodies. That's also very exciting and interesting domain. And we were able also identify the potential position of the mutation, which leads to desirable, let's say, unfolding temperature, aggregation temperature, or even interaction parameters, which is a very essential information when you're building your vaccine or targeting certain target or disease. And a couple of more applications obviously related to image processing and application of the machine learning towards the analysis of the images. And we've done a number of projects, including one which is, related to analysis of the gel images. And that's something, very interesting to see how the machine learning can bring very important results in discoveries. And last but not least, obviously, reanalyzing database or using the machine learning as a quality control check of the potential results. That's something we also able to do and have a lot of successful project in the past. And I think I will end my presentation here. And if you have any question, I will be very happy to answer them.
Thanks a lot, Sasha. That was great, and thanks, guys, for your talks. Yeah, please put your questions in the Q&A or in the chat box. While we're waiting for this, Sasha, I was just curious on that repurposing of targets. What type of what's the range of success there? Like, because obviously, you have older tools, older data, and they may miss things. Now you're coming back at it with a fresh approach. What are you seeing in the industry when you do that?
Well, I mean, obviously it depends on the application and the data in hand. So let's say if we are talking about protein engineering, so when we do the prediction, we give us a confidence interval or probability of belief that our prediction is a confident. And you can see it all depends on the model you build it, and it's very important, aspect of machine learning when you build confident model. And then most of the time, in 85%, 90%, if you build a very good model, you can confidently predict the results. It doesn't mean that you cannot get false discovery or very low confident predictions. But that's why very important, not just make a prediction, but also provide the confidence interval next to that. That uncertainty analysis and quantification plays a very important role. And it's very important also for other applications, even if you're using conventional bioinformatics pipeline and you use the machine learning as a quality control, you can see sometimes, how consistent you are with your logic or criteria behind the analysis when you just, basically use the machine to see if you can learn from that analysis, what was the logic behind. And that's very important as well. So in that sense, the answer is maybe not very direct, but it all driven by replication. But you could have, you know, very successful story and not so successful story, depending what you put in the first place.
Right. It does sound something that could pay off, though, for somebody that says, hey, we're going to go apply this to some older data that and see if we missed anything, right? At a low cost, low overhead.
Yes.
And you don't know, you might, something might come out of that. So that's kind of cool. Okay. Hello. Amazing presentation, short and concise. My question is, what is the success rate of the entire procedure in real world problems? It's kind of similar to what I was asking. How many such approaches have actually made it to clinical trials?
So maybe I can jump in, for example, that presentation about the protein gene goes via validation stage with one of our clients. So we don't know yet what would be the outcome, and we are kind of very interested and looking forward to see those information. But let's put it in terms of the prostate cancer diagnostic project. So we have the machine learning capabilities allow us to get up to 90% sensitivity specificity on the set of the biomarkers the client developed. And it's much better compared to the standard PCI test, which is exist in the clinics, you know, which leads to a lot of false discovery and mislabeling of the sample. They have a specificity about 40 to 45%. And it's a very big changing technology. And even further down, so diagnostic between medium level cancer, which, you know, can be monitored without any intervention of the biopsy, versus a very highly aggressive cancer, which requires a lot of intervention. That's very important, because you don't want to put the patient into the subject of doing the biopsy and then, you know, some complications result of that. So
this is actually really critical, Sasha, because we're all getting to older and stuff, and you hear a lot about prostate cancer today, and you actually hear what you just described. People are like, it's super slow growing, you don't really have to worry about it. And then you hear, well, that's what they thought at first, but now they've changed it. It became aggressive. There's a lot of what seems to be somewhat understood, not well understood. And a lot of, like you said, there's a lot of variety that becomes very confusing to the patient. And I think this is really powerful.
And that's why you need to have a very good advanced system in place to make those recommendations and provide, you know, appropriate guidance what needs to be done with the patient, right? Including the monitoring, check the biomarkers, you know, and then make the prediction how the disease is evolving. Yeah.
No, that's really good. From a Regulon perspective, Emil, how new is this analysis? How long has this been going on in the industry?
Well, it's all quite novel. If you consider that single cell sequencing only starts taking off 2015, so right. That's really your baseline level when you're starting here. CNIC itself is only, let's say, 3 years old, with the new wrapping, let's say, the algorithmic optimization. That's really just last year that's really being published. So in terms of how much is this type of analysis contributing to clinical trials, at this stage, we're mainly in the preclinical phase for CNIC. If you look at the citations of the original publication, in just around 2 years, you're talking more than 600 citations. So people are looking at the tool, people are using the tool. So I hope to have more numbers for you 2 years down the line or something.
No, that sounds great. Well, with the onslaught of multiomics and the critical, there's some critical things here with multiomics. And one is not only the data contextualization, the data storage, the data curation, but then eventually the reaggregation of all that data and trying to figure out how you can bring it together to make obviously better decisions and stuff. How are you guys approaching that today? How are you making sure that your data is going to be reusable?
Maybe I can add. Oh, sorry.
Sure. Go Sasha, go ahead. So I will add.
All right. Sorry to interrupt you, Matthias. So I think it's a very good question, especially in the world of the big data. The single cell analysis on its own is a huge topic, and it has a lot of potential. Then machine learning and some other, let's say, you're trying to combine imaging, single cell analysis. and some other things, clinical data, which is, really coming from different worlds together, but building that into one predictive approach is, what we are targeting, because that's, allows, to extract information from all sources and then make a right decision. And I think, what the industry is moving towards using AI for that type of application, using the knowledge graph, doing AI on the graph, where you have could be completely unstructured data, put it on the graph. That's something, where we're moving towards. It's going to take some time because agglomeration and structuring the data and then developing the right tool, which takes all this huge information into one computational framework. It's kind of state-of-the-art problem, but we are getting there, and then there are more and more examples coming up, including what we do in the BISC. We do combine some of the machine learning activities, as actually Emil showed in his presentation, random forest model as a part of the machine learning algorithm, which he utilizes to make certain discoveries, right? And obviously combining with the cloud technology, that's also an essential part of making that computation fast, robust, and you can agglomerate it everything in one pipeline.
Yeah. Well, it sounds from what you've described, you're getting incredible outcomes on your machine learning. Someone like me in old school, the way we used to do a long time ago, we weren't getting 90% predictability. That's amazing. So you guys are doing something awesome and right. What's next for BISC Global? Like, what's what's your goals to achieve next year?
I think that's probably a question that I will jump in. First, let's take a look back. I think we've been doing very well over the past couple of years, knowing that we've only been around for a little over 4 years now. I think in the previous year, we pretty much doubled in size as a company. For next year, that growth is expected to continue. So both in terms of the consulting work we do, the projects we do, we are expecting to increase that, also close to doubling is the intention. On top of that, we're doing some internal development projects as well, meaning we are developing a couple of softwares, particularly for data analysis, which currently have been pre-commercial phase, let's say, but as they progress, the goal is also to start pushing those a little bit more and actually start offering those to customers as well, somewhere throughout 2020.
Yeah, no, it sounds great. There's a lot of buzz around you guys. What we saw today was very impressive. Is there any way to leverage like some of the DeepMind work that's been done in your protein prediction or protein analysis or anything? Is there any tools that are coming out that have been open source where you see you might be able to leverage things like that in your work?
Not at the moment in terms of the open source tools, because whatever we are doing for the client is IP belongs to the client, because that's their treasure and asset, right? In terms of identifying set of the mutation position and building the libraries. Effectively, that's, their product, if they're targeting, let's say, antibody treatment, type of technology, right? Or in some other cases, the CFI deficiency disease, is also very important. So making the mutations and increasing the AC50 activity will lead to successful results in treatment. So from that point of view, we are not able to reveal that information, but maybe at some point we will definitely come up with the publicly available tools, or maybe commercially available tools to address some of the community interests.
Yeah, no, it's awesome. I was just curious if like DeepMind if there was any way that could be pulled into some of the workflows or what you're doing around your predictions, etc. But it's very impressive, and I think the world needs as much biology analysis as possible, right? Obviously, so much has been done in the small molecule world. We still don't, there's still a lot to do, a ton to do. But in the large molecule world, there's still a ton of help needed, right? And it seems like you guys are really going down that path and making some big differences. So it's really good to see that. Any comments from any of the speakers or anyone else before we call it a wrap? And yes, this is recorded. This will go on demand and you'll be able to access this later for that question. All right. Thanks, everyone. This has been great. I think what you guys have done is awesome and fantastic, and look forward to working with you next year on continuing your success.
Thank you.
Thanks,
Thanks,
John.
everybody. All right. Take care.
All right.
Bye.
Take care.